Right. So, all right, is Anna here? She is next up. Anna. There she is. Come on up. Don't be shy. So, Dr. Anna Pfalzer is a research assistant professor of neurology at Vanderbilt University Medical Center. Her research identifies early biomarkers of neurodevelopmental and neurodegenerative disorders with an emphasis on rare genetic conditions. She holds a PhD in biochemistry from Tufts and completed postdoctoral fellowships in neuroscience and pediatric neurology at Vanderbilt. So take it away. Anna, is that? Should you just click forward? There we go.
Okay. Okay, that's close enough, I think. Okay, thank you, Eric, for the introduction. It's wonderful to meet all of you guys today. I want to talk a little bit about the work that I have been doing. This is mainly a project that was run through Combined Brain, where Dr. Bichelle is going to speak with you right after me. But this is a project that is looking for biomarkers of rare genetic neurodevelopmental disorders such as Kleefstra syndrome. So one of the first things that I wanted to just briefly introduce is the concept of what is a biomarker. And this is a term that gets thrown around quite a bit, but it's a pretty straightforward way to explain it, is that is a biomarker is an objective measure of your kids or young adults or adults' disease status, whether they are progressing or not, for example, and they can also be used to predict an individual's response to treatment. These, more importantly for community, these tend to be more sensitive to different types of intervention, and what I mean by that is they tend to change more quickly than things like outcome measures of cognition or memory or communication or speech. So it's really something that you can use to measure whether an intervention like a drug is working before you'll see changes in sort of cognitive or behavioral profile. So biomarkers are typically things like different genes, different changes in DNA, different proteins that get produced by your body, metabolites, or things found in your gut, like microbiome, like gut bacteria. And because of those, you find that biofluids. Can you see my little red marker thingy if I squiggle it around a little bit? Okay. You find biofluid biomarkers in things like cerebrospinal fluid, so the fluid that bathes your spinal cord and your brain. You also find it in blood, urine, saliva, as well as everyone's favorite is finding biofluids in stool samples. Now, one of the things that's really difficult about finding a biomarker for a neurological disease is it's really hard to get into the brain to see what's going on. So it's hard to find out and get access to fluids that can recapitulate what's happening in your brain without actually getting access to your brain. But this has been done successfully in other neurological diseases and even rare genetic neurodevelopmental disorders. Just for the sake of time, I will not explain really in great detail these figures, which you probably can't read the figures on the graphs anyway. So for this project, I had a couple big questions. So this was a project that was across 27 different neurodevelopmental disorders, and for a rare disease group and a pilot project like this, I really had two primary questions I wanted to answer. The first one was, does the blood from Kleefstra syndrome patients, does it look very unique to all of the other genetic neurodevelopmental disorders I collected samples from? Or does it look really similar, even though they're two very genetic, distinct disorders? So this is what we did. This is how we collected samples. We tried to get 20 patients from each disorder in this project. And I would venture to say that probably a handful of you in this room had your child donate for this project. So I apologize if your child sees me and starts to cry. I did that quite a bit in the last handful of years. And then we also collected from neurotypical controls, which is a theme that you guys will probably hear repeated over and over again, is it's hard to tell what is different in a KS patient if you don't have something to compare it to. So siblings don't like having blood or other fluids collected, but it is really a critical part of a project, especially a biomarker project for rare diseases. So we collected a tube of blood from KS patients as well as a handful of other disorders, and then we had everybody fill out their favorite neurodevelopmental assessment, which is the caregiver-reported Vineland. I understand that everybody loves doing that, and we have copies in the back if you have not gone through PTSD enough already. And then we did this SomaScan proteomics platform, which just measures all of the proteins that you can find that's circulating at a blood sample. Okay, this is a very complex, busy table, but the important stuff I pulled out over here on the left-hand side, where we collected blood from 18 Kleefstra Syndrome patients, and we used 18 controls that were matched by age and sex. You can see the average age of Kleefstra Syndrome patients was 15, but we had a pretty large age range in those donors that donated to this project. About a third of patients reported epilepsy, and they all, based upon their Vineland scores, had somewhere between moderate to severe intellectual disability. So I'll show you 3 of the little bits of this study that I think is particularly interesting. So one of the most important findings that we have discovered, or at least we have preliminary data from this project, is that the blood and the protein circulating in the blood of Kleefstra Syndrome patients does not really look much different from other genetic disorders when you're looking at sort of a high level. And this sort of like mishmash of different colored dots is kind of what's proving that idea, as each dot is an individual with one of those 27 disorders. Purple dots are the KS patients. And without really squinting your eyes to try and see which disorder is which disorder, you can get an appreciation that the colors are all mishmash and jumbled together, right? The purple dots for Kleefstra Syndrome patients don't all aggregate together. So this is really surprising and interesting from looking for shared biomarkers, right? That is something that we in the rare disease space are really looking for. Are there shared biomarkers that we can use across rare disease communities so we can stop thinking about them as just treating one disorder and treating one disorder and treating one disorder separately? This is another way of looking at the same data set. If you pay attention to the figure on the left right here, you can see what you might have already suspected from knowing other rare disease communities is that Kleefstra Syndrome patients, that's this yellow highlighted phrase right here if you can't see it, the blood from those patients looks really similar to a handful of other chromatin modifying disorders, which is Coulon-de-Vries syndrome, Kabuki syndrome, and CHD2, these are all known epigenetic or chromatin modifiers. So this kind of makes sense and is really encouraging if we're wanting to think about maybe studies that could be beneficial for a handful of rare diseases, not just one at a time. Okay, those weren't supposed to be there, so I'm going to keep going past that. This is the last slide. So I looked at samples from a lot of different disorders, but what I want to focus on is just what are specific things that we think are happening in the blood of Kleefstra Syndrome patients. And without going into a lot of detail, we see about 100 proteins that change in the blood from Kleefstra Syndrome patients to those neurotypical sibling controls that donated. And when you look at those 100 different proteins, you say, okay, that's great, that's exciting, there's a lot of things that are changing, but how do they sort of come together? What do they all sort of impact? And it seems like the answer is really circling around things that are important for neuronal differentiation or neuronal growth and survival, as well as neuroinflammation. And that is probably a trend that you will hear in several other talks is the role of inflammation in Kleefstra Syndrome patients. And these are a handful of proteins that really are involved in those neuroinflammatory, neurotrophic pathways that we found that are enriched and potentially important in Kleefstra syndrome. So this is the last slide that I will leave you with today, which is showing really the essence of a biomarker is saying what changes in Kleefstra syndrome that doesn't happen in a kid that's the same age and the same sex. And the holy grail is really a biomarker that changes with disease severity. So something gets higher, for example, as the kid progresses and gets more severe. And this is an example of what we find is an exact sample of that for Kleefstra syndrome. So here's a particular protein, GDF15, and the levels of that protein increase with increasing communication ability as determined by their Vineland. So this is exactly the type of thing that would be a piece of preliminary data that we could potentially validate as plasma GDF15 as a biomarker for Kleefstra syndrome. Okay, so conclusions from today are that we do see that there's some shared plasma proteins that occur in Kleefstra syndrome patients as well as other rare genetic neurodevelopmental disorders. For the sake of time, I skipped talking about epilepsy, but we do find that kids who have epilepsy, the elevation in the proteins that we found are even higher in kids who have recent seizures. And then we also find some proteins that are specific to Kleefstra syndrome, and they seem to match what we have seen in other preclinical models about neuroinflammation being a key mediator of the disease. Okay, so here's where I say keep having your kids donate various biological samples when you can. Because even though this is exciting and really promising data as a biomarker for KS, we need additional samples. So if you haven't donated, adding to our sample size would be great. And even more importantly is we need longitudinal samples. So like every year, every 2 years, it would be great to see if these changes in some of these targets that we have found are validated and remain elevated. And then also, it would be really great to say, yes, the things that we find in the blood also are found in the cerebral spinal fluid of patients, because that would really convince us that it is something that's important in the brain. Okay, so this was a massive undertaking. This was a project done by Combined Brain. Here is everybody who had some hand in this project. It was led by Dr. Bichelle, the current CSO, Lindsay Wazorek. Data sharing was done through Simon Searchlight as well as RareX. Ricardo is our computational guru, Natasha Ludwig for her help understanding Vinelands, and Victor and Wayne for their help with proteomics. Okay. Oh, right, of course, you guys. Thank you to all of these foundations who helped recruit for this project. Quite literally, I would not have been able to do it if I hadn't continued to harass Jeff and Eric for finding KS patients. So, okay, thank you.
Any questions?
Thank you very much for the talk. It's really interesting. You mentioned that plasma levels of the GDF-I5 significantly correlated with receptive communication skills. You'll have to forgive my statistical questions, but that was how many correlations, and could you tell me something about the effect size and the confidence interval as well as the significance level?
Okay, so are you saying how many proteins did we test to find that one image that I showed you before?
Right. Yeah.
Good question. Okay. So on the slide before that, I have no idea if I can go back. Let me try. Okay. So we did this 100 times. So we said we found that there were essentially 100 proteins that were different, up or down in KS. And so what we said was, from some preliminary work, we knew the specific variable that we wanted to look at, and that was communication from the Vineland. For a variety of reasons, probably which I could not explain, different aspects of communication seem to correlate more tightly with some of the biomarker studies that people have been doing. So we specifically picked out communication as our variable. And we did more or less 100 correlations. We say, which of these 100 actually correlate with a clinical marker of severity. And actually what I blew right past is that it wasn't just this specific protein. This is the one probably realistically just had the prettiest graph. But we also found that 25 of those other proteins that were differentially expressed also look exactly like this.
Okay, any other questions? Okay, we'll move on to the next speaker.